Hello, and welcome to this video on genome editing. Genome editing is a rapidly advancing field of science that holds the potential to transform the way we approach treating genetic diseases and improving agriculture. In this video, we'll explore what genome editing is, how it works, its potential benefits and risks, and some of the ethical considerations around its use. At its core, genome editing is the process of making precise changes to the DNA of an organism. DNA is the genetic code that makes up all living things, from bacteria to humans. Genome editing allows us to manipulate this code, making it possible to add, delete, or modify specific genes. This can be done in a variety of ways, but the most well-known method is known as CRISPR-Cas9. CRISPR-Cas9 is a gene editing tool that uses a combination of a guide RNA and an enzyme called Cas9 to cut DNA at a specific location. This allows researchers to add, delete, or replace specific genes. The process works by introducing the guide RNA and Cas9 enzyme into the cells of the organism being edited. The guide RNA helps Cas9 find the specific location in the DNA where the edit needs to be made, and then Cas9 cuts the DNA at that location. This triggers a repair response by the cell, which can be harnessed to either remove or replace the targeted gene. The potential benefits of genome editing are vast. For example, it could be used to treat genetic diseases by correcting the underlying genetic mutations. It could also be used to create crops that are more resistant to disease, drought, and pests, potentially increasing food security around the world. Additionally, genome editing could be used to create new medicines and therapies, such as cancer treatments that target specific genetic mutations. While the potential benefits of genome editing are significant, there are also potential risks to consider. For example, unintended changes to the DNA could have unforeseen consequences, such as creating new diseases or causing harm to the environment. Additionally, there are ethical considerations around using genome editing to modify the DNA of human embryos, which could lead to unintended consequences for future generations. The ethical considerations around genome editing are complex and multifaceted. While there are potential benefits to using genome editing to treat genetic diseases, there are also concerns about using it to modify the DNA of human embryos. This raises questions about the potential for unintended consequences and the implications of modifying the genetic code of future generations. As such, it's important that the development and use of genome editing technology are guided by ethical considerations and transparent communication with the public. In conclusion, genome editing is a rapidly advancing field of science that holds significant potential for transforming the way we approach genetic diseases, agriculture, and medicine. However, there are also significant risks and ethical considerations that must be carefully considered as the technology continues to develop. By working to understand and address these concerns, we can help ensure that genome editing is used in a responsible and beneficial way.